Okay, let's start with the afternoon session of today. So today, the topic of this afternoon will be how to perform genetic association analysis on the lifelines data on the UMCG high performance clusters. And for this purpose, I had invited Esteban Lopera Maya and Robert Warmerdam from the genetics department, to share their experience on genome-wide association studies on the UMCG high-performance cluster with us. So Esteban, please go ahead. The floor is yours.
Okay, thank you very much, Lydia. Good afternoon, everyone. My name is Esteban Lopera. My partner is Robert Warmerdam. We're both PhD students at the genetic department. And so today we want, like, a big chunk of our work involves using the genetic data of lifelines to make genome-wide association studies. So we want to share that with you and like give you a little presentation to give you some elements on how to do it by yourselves. Let's see if this continues. Yes. So first a little outline. I will try to give a little introduction to remind the theoretical part of the genome-wide association studies. Then we will dive into the data structure and status. We'll see that it's very important that you know how's the data structured. We'll see some of the problems that you will have to face, and some of the software of the tools that you will use to overcome it. Then my partner Robert will give you some insight into more efficient use of the HPC to parallelize methods, some meta-analysis, then quality control, and some of the downstream analysis. Yeah. So genome-wide association studies are designed to detect an association between alleles or genotypes and a phenotype or a phenotype that can be a disease, a binary disease, or a phenotype or a quantitative trait. Sorry. So it's a very simply just a regression model that explains the phenotype by covariates and the genetic variants. But as you know, we do this with not only one SNP, one genetic variant, but we do it with as much as we have a genotype or sequence, depending on the method. And then we're going to do this regression many times. So this is thousands or millions of SNPs, and that means we need to correct for significance, and usually the expected you correct by the expected number of tests, which is the size of the human genome, so 3 billion, and that's how you get this p-value threshold for the standard significance. Now, usually when you find something significant, it's not considered real until you can replicate it properly into in an independent cohort, or if you use a meta-analysis and you find it again. And also, you need to have in mind that the SNP, the variant that you find significant, is rarely the actual causal variant, but it's rather segregating together with the actual causal variant. So you need other methods to actually detect the causal variants. And well, those methods are not going to be part of this presentation, but it's good that you have that in mind. So for the data, so for lifelines, we have two sets of genotype data. First of all, we have UGLI, which is a genotype for 36,000 individuals. This one is in two forms. First, we have the hard-coded genotypes, which is just the direct output of the genotyping array. This is about this number of SNPs. And this is in link format. So I don't know if you have heard about link format at this point. This is three files, the .bin, which contains the SNPs, the .fam that contains the samples, and then the well, the SNPs or the variants, and then the .bed, which intertwines them together. You can easily see the .dim and .fan. They are readable by humans, but the .bed is binary file, so you cannot really read it. It's not intelligible, but machines do that. So this is how a FAM file would look. It has the information of it has a family ID, individual ID for each sample, and then sex and phenotype. This is, of course, all in zeros, so you really don't see that information here, but you could. The second kind of format that we have this data in is the imputed genotypes. So this is, we take the genotypes and we take the genotype data and we consult these variants in a reference genome that has the haplotype information of these variants in common in known population, and that will give you then a guess of what's the rest of the SNPs according to the variants that you have, and then that would increase the number of variants that you have. So in the case of UGLI, we increased by 40 million of SNPs, and this data is we have it in three formats: BCF file, which is readable by human, BGen and GDS, which are not. And this is how a BCF file would look. Important here, I wanted to show you, you have the information of the frequency, in the panel, the reference panel that we use, and you have the information score, which is like the reliability of the imputation, so you know how much you can trust in this SNP. And then you have the genotype for each sample in different columns, which you see here as a lot of numbers because it comes in three different formats. And I want to talk about these formats a little more because you might need to know this. So you have first, it could be shown as the genotype, the direct genotype format, where each allele, both parental alleles are represented by a number that can be 0 for the reference allele or 1 for the alternative allele. And so an individual that has paternal allele 0 and maternal allele 0 would look like this, and so on and so forth. So this is the genotype, and then you have the SAIGE format, which what it says is it's a continuous number that says how much of dosage of alternative allele you have. So can be 1 or 2, but it can be also be 1.2 or 0.6, I don't know. It's a continuous number, so that's important because it gives you more precise reading. This is a continuous number. I know it's kind of difficult to grasp why, but that's because this is calculated from the genotype probabilities, which is the third format, which is just how much of a probability of each of the possible allelic states you have. then in theory, they will sum up to 1. Yeah, and different softwares will use differently these 3 formats. It's good that you know this. So as the data itself, before you work on it, you need to know a little more of it. So first you find it in this location. It has all the files are is separated by chromosomes. So you have the autosomes and the X chromosome, and this is the pseudo-autosomal chromosome, and it's only available for the genotype data, but not for the imputed data. The genomic positions are based in the human reference genome version 37, which is not the latest. So you really need to check this if you're comparing your results with other studies or databases, or yeah, mostly when you need to do validation, you have to have this in mind. The family and sex information is not included in the genotype files, as I showed you a couple of slides ago. The variant names are not all RS codes. So you have some RS codes, but you have also things like this, or you even have things like dot or dot dot. So that's something that you have to take in account. Always try to look for the position, reference, and alternative allele, and that would be the single identifier of the SNP. And lastly, and very, very importantly, we noticed after we finished imputation, this data, we noticed that this data has some discrepancies, some differences with the reference that we used for imputing. So difference in like reference allele notation and the allele frequency, and then that these SNPs cannot be used because they are not reliably imputed then, and so they need to be removed from the data, but then that has not happened, so the data actually includes them. So if you're going to work on this data, I really, really recommend you to look for this file, which has the names of the of these SNPs, and remove them yourself after your analysis is finished. So we also have a little about, I want to say 15,000, I think, individuals that are genotyped with a different genotype array. They are located here. And we also have hard code genotypes and imputed genotypes. This data is also phased, so we have Oxford genotype also. And yeah, this is kind of a share kind of the same characteristics. Okay, I would like to know if there is any question up to this point. Okay. In that case, we'll move on to some of the problems to overcome. So the first would be families. As you might know, Lifelines contains up to 3 generations of families, which results in and can result in an inflation of allelic frequencies. I mean, so you will have more representation of some variants just because you have people that have the same variant that just come from the same family, which sense that gives you inflation, and that can give rise to association that is not real. It gives you false positives just because there is more concentration of some SNPs. So that can happen, but also you have to be aware that this relatedness is not always obvious by family structure. So you have also like second cousins, third cousins, or people that they don't even know they are related, and they are going to also share this this higher quantity of similar variants, and there is no way to know from the family structure. So what you use to detect this is you measure the, let's call it the genetic similarity. That is basically the frequency of shared alleles that can be, usually you find it in the test like identity by descent or identity by state. But for this presentation, we just call it the genetic similarity. And then you can use that and as also the family structure to just define a threshold or where to remove people from your study so they don't inflate your variant frequencies. So that can be done in the most simple softwares. But what's recommended, the most accepted way to handle this problem is just add a genetic relationship matrix, which is just a matrix with the pairwise calculation of this genetic similarity, and that can be added into the model, maybe advanced in a way in a linear mixed model, which takes this as a random effect. So it's a special model for cluster data, just as this. You don't really have to care much about that because the software usually just has it by default and does it by yourself. Yeah, so that's the two ways that you can use to handle this. We have the familiar structure is located here, if you're curious about it. And the genetic similarity matrix, you have to calculate it yourself. Well, we also have it, I will show it later. Second problem I want to talk about is population stratification. So this happens when you have individuals from different populations in your data. As you know, different geographical regions that they are very diversified by ethnicity, can also be diverse by other covariates, but mostly the ethnicity means that you will have different linkage disequilibrium structure. It means that the way the haplotypes are distributed are going to be different from one person to another. And that will give you also false positive, but also can also mask real associations. So you really want to avoid, you really want to avoid this. For the data in UGLI, that's not a big problem because they are very little. And how you measure this is you do a principal component analysis. Principal components are an eight-dimensional way to measure the variability of a data set. In this case, it would be the genetic data. So if you look here, for example, this is actually UGLI data directly. The brown triangles are the UGLI data, and the other dots are just individuals from 1,000 genomes, superpopulations, so different populations. And you can see that most of the UGLI data is clustered together with European populations of 1,000 genomes, and then the 35 or so outliers are actually clustering together with other populations. Here you see them with Asian and stuff. So once you know this, once you have the measure, you can, to control it, you can either remove these population outliers, and or add the principal components, at least the first ones, to the model. So most people use up to the 4 first principal components. Some people use all 20 principal components. Usually both practices are recommended because you still have some population substructure even if you have only one population in your data set. So yeah, this is just very recommended that you do. You have the principal components for the ODLI data located here, and you also have the individuals flagged as non-Europeans located here. Yeah. I have a question. I think I should answer it now. Go ahead.
Hi. Yeah, I'm just curious about the 35 individuals from non-European. I was expecting that during the QC process of the genotype data, this makes sure that we do have only Caucasian samples or European ancestry, and then how this can at this stage.
Yeah. I shouldn't have this data, but I actually have it because I was the one who managed the quality controlling ugly data. so I can answer that. We decided to, instead of removing them, which is the usual practice, we decided to keep them because because we thought that they would be enough to make sub studies. This was not the case; they were too little. But then we decided to leave them flagged, so you can remove them by yourself or include them if you feel that they make no difference for your research question, because it's also a matter of how important is this for the research, how much this is going to change. So, for example, I study microbiome, and you can imagine that the microbiome of people that lives here, it depends a lot of on the diet, would not be that different depending on the ethnicity necessarily. So, for example, those individuals I did not remove for microbiome studies, but for other studies, it's important to remove, and then this is why they are still there. But then you have them flagged, so you can remove them if you want.
Okay, thanks. Anyway, yeah, you made it simple for us during whatever to the decision to remove them or still.
Yes.
Yeah.
Yes, it's better to leave it to the researchers.
Yeah, thanks.
Okay. So the next problem to overcome would be case control imbalance. So we have a large cohort when you look at this data for quantitative traits, because you have 36,000 samples. But when you try to look at the disease, then it gets tricky, because then you can have diseases that have sample size as little as 200, 150 people. So that's going to be a problem. This can lead to something called case control imbalance. When this is too high, you're going to have violations of the assumptions of the logistic regression, mostly homocelasticity. You're going to have different variance across the different covariates. And that is going to give you high coefficients estimates. Also, you can have a separation, like a small. If you are picking such small groups, you increase the risk of grouping together all the variability of the model. So sometimes you can even get probably an error message saying that the regression model does not converge. Yeah, that's going to happen sometimes. So there is really not much you actually can do about it. What you can do is keep in mind that the sample size would be enough according to the number of covariates. So, for example, if you have 50 individuals, do not have more than 5 covariates, something like that. But even then, you're going to have a big case control imbalance, and that's going to give you some false positives. So locally, there are corrections that are very mathematically intensive to discuss here, but they are already implemented in many of the packages that we use. So there is a field regression, which is going to weight the height estimates of the coefficients. So it's going to make them less the effect less tolling. And there is also some of the linear mixed models also include score tests that will take in account the case control imbalance and will very much remove this problem. So the main software that we're using now does that, so that's good. Okay, so I'll talk a little about the tools that we use for genome-wide association studies. The first one I want to touch on is Plink. So Plink is the most important, most useful, I would say, because it has a very complete array of tools, not only for GWAS, but for all genetic data management, like frequency calculation, quality control, etc. You can build them yourself. You can modify yourself. So it provides a lot of flexibility. If you're going to do GWAS, even if this is not going to be your main software, you're probably going to be using it just to process data. So it's very good that you take a look at the manual. That is probably here. For GWAS specifically, you have two main functions. That's linear for quantitative phenotypes and logistic for binary phenotypes. I think there are more, but this would be the main. I want to mention a little the rest of tools, because of course there is many, but they are going to have just different approaches to the most of the problems that we already mentioned. Plink does not correct automatically for any of the problems that we mentioned, but as I mentioned, you can do it yourself, including the model PCs, remove outliers from family, remove family members, remove outliers from PCA analysis. So it's still like you can still work with it, but you are not going to have all these advantages. GCPA has a very good distinction for family and maybe for cryptic relatedness too, but it still removes and calculates apart the relatives. It's very good to calculate heritability, but you do need to feed the familial information. HEMA and GMAT is the first one to include the linear mixed models. So it does try to control cryptic relatedness, which is very good. And then you have bold element, which improves the algorithm in GMAT. It's more efficient, and I think it includes also more precision for binary traits. It starts to use the saddle point approximation. Adds the score test, corrects for population stratification. SAIGE, which is our main tool now, improves and uses all of the algorithms that we mentioned, but it has a very good efficiency and precision. It also, well, it corrects fully the case control imbalance. So this is why we use this, but it's not only designed for binary traits. I want to stress that because most people think that that's the case, because it corrects for this. Then there is last year they released Regini, which improves in of the previous algorithms also, but adds option for different packages, like different kinds of analysis, I think, like burden test and gene-based analysis. It's also designed mostly for parallelized computing, so it's extremely quick compared to SAIGE. I think, no, I don't remember the number, but it's definitely faster than SAIGE, and it handles differently the genetic relationship matrix. Yeah, it's just more efficient with it. I myself haven't used it. If you're willing to try, I encourage you to do it. But we do not have any structure yet. Like nobody has worked on it yet in the cluster on Gearship. So yeah, if you're going to try it, you're kind of by your own. Yes. Ilja?
Yeah, so can Regini, can that also be used with PCF files? So version any?
Oh, yeah. I actually not sure what's the input for Regini yet, but definitely it's not. You do not need the GDS conversion because the GDS conversion is unique for SAIGE GDS. Is that the question?
Well, yeah, but yeah, the question was if Regini can use VCF files. But
yeah, I'm not fully sure.
Robert, maybe you should know.
Yeah, I've looked at that, and it appears that Regini can use both BGN files and Blink1 and Blink2 version files. I'm not VCF files. I'm not seeing on the list.
Okay, but then Blink files you don't have this these genotype probabilities, or can it use the dosages?
No, Blink1 shouldn't be able to use it, but Blink2 definitely can.
Yes.
So that it's a disadvantage that you lose these genotype abilities.
Yes.
But SAIGE can use the imputed and accounts for the imputation inaccuracy.
Yes.
Yes, but as said, Regini also accepts BGN files and Blink2 files, and those definitely can handle with dosages.
Oh, okay.
Yes, yes. Okay. So now I'm going to focus a little on the main tool that we use for now. So as I mentioned, it includes algorithm of all the others. It uses a genetic correlation matrix, and for efficiency, it works in 2 steps. The first one, it's going to fit the null model to estimate the various components to the phenotype. So it's going to take the phenotype, the correlation matrix, and then do a regression with them together. And the result from that is going to use it in the second step for test for association with each of the genetic variants. So in the paper, you will find that they do not recommend it for sample sizes of less than 500. I have used it with sample sizes as low as 200, and it still works, but it has a higher probability to fail. So I do not recommend it to use it for sample sizes less than this, but it could work. There's uncertainty. Oh, well, if that happens, you can use Blink. You can always use Blink2. That would be my to-go option. Yes. Yeah, the last thing is, well, you will notice I have been mentioning two versions of SAIGE. So We have SAIGE GDS, which is a software that is designed for sequence data. We do not have sequence data for the time being, but it can also process the data that we have. It has the disadvantage that it's going to use the genotypes. So just as Ilya mentioned, it's not going to be very precise, but that also makes it very, very quick. So you will have a full association of one chromosome in 20 minutes, more or less. What else can I mention? The updating is not very active for this one. I have not seen an update in a while, and well, it gives a lot of false positives because, well, a bit of false positives because of because it uses the genotype. But this is easily corrected by genomic control, which Robert will tell you about this later. Works within R, which is usually good for people who's not very familiar with coding, because R is kind of easy for most of us. Then we have SAIGE, which is an improvement in some ways, because it can actually use the dosage information, so it is a little more precise when calculating associations. But then it has the downside that it uses 2 to 5 hours per chromosome. It works in BCF or BGen files. It's a little faster in BGen files, I seem to remember. It has an update that is very, very active. I think 2 months was the last version that I noticed. And it has higher precision. I have not used actually genomic control with results from SAIGE, from this SAIGE. I don't know if Robert has. And it works outside of, so it works within R, but you call it outside of R. Yes.
So we previously tried to use SAIGE on the cluster, but the manager said that they couldn't install it. But you were able to install it, or?
So which managers? You mean Lifelines data managers?
No, from the GCC.
From the GCC?
Yeah.
This was how many years ago? Or
a year ago, when we just started with the ugly Lifelines GWAS.
Oh, weird. Okay, so right now SAIGE is working in a framework that is called oh my god, it's not Docker. What's the name of it, Robert? Do you remember?
Oh, Singularity. Yeah, we use a Singularity container.
Yes. So yeah. So I agree, it's very difficult to install if you don't use Singularity, but then they made it available by Singularity. I think we made, so we have this, maybe I can try and take this link. We made this GitHub page. This is not the right page. Can you correct the link, Robert, when we when we deliver the presentation?
You want the link?
Sorry for this. Yeah, do you have the link, maybe?
Yeah, I have the link, but I propose to move on because there isn't much time left, I think.
Oh, I'm very sorry. You're right. Okay. Yeah. Okay, but the thing is, we have a GitHub setup that you're going to use, I think, for the workshop. And there is also SAIGE GDS, but there is also SAIGE, and you can use it from there. I think my presentation is has been messed up. Yeah, thank you, Robert, for putting the. You can see the link in the chat, and if you follow, there should be instructions to use both SAIGE and SAIGE GDS. I'll just continue like this, if you don't mind, because I'm almost finished with my part. So to work in SAIGE, you need BCF files or BGen files in SAIGE, GDS files in SAIGE GDS. You need the phenotype files, and you need a list of. Oh no, this is really small. So small. Yeah, and you need the list of covariates. You need to have your data converted to GDS. That's a bit of a problem. We do have already the oddly data converted to GDS. You also need to build the genetic correlation matrix. You can do this with GDS within R. You can also see that in the GitHub. For SAIGE, you can use Blink, just do a normal in-depth function, this code. And we have already built a genetic relationship matrix for it. You can find it here. So once you have that, you just do these 2 steps. This is just in the console in leadership. Oh no, sorry, this is within R. You define the model, the phenotype file. You give it the genetic correlation matrix. You define the type of trait. If it's binary or quantitative. It's recommended that you conserve the inverse normal transformation if you're handling a quantitative trait. And for the step 2, you just take the result from this, and you have the option to do the association only on the SNPs with certain filters for information score or for minor allele frequency. Yes, that's.
Thank you, Esteban.
Yeah, sorry if I took a little more time.
Okay. Can you see my screen now?
Yes.
Okay, great. So I'd like to go into the performance and parallelization a bit using SLURM. So as Esteban alluded to earlier, SAIGE GDS and SAIGE require different amounts of time in order to run. So for example, 20,000 samples in SAIGE GDS can be completed in under half an hour, when for a single chromosome, well, SAIGE at least takes a couple of hours. Both, however, require very little memory. And I would recommend to experiment a bit with what you request from SLURM to fit your workload nicely. Chromosomes offer a great way for parallelization, since you're able to assess every chromosome separately. One way to parallelize using SLURM is to submit a separate job for each chromosome. However, a bit of a less naive way would be to use the array job functionality that's present in SLURM. And using that, you can manage your job as if it were one, or all the separate chromosomes are running on different nodes on the cluster. So that gives a little bit of an advantage. And we have also implemented this in the bit of code we have prepared for the practical session later on. So as Esteban mentioned, there are 2 genotyping platforms. When you want to include both of these, you should use a meta-analysis. And when combining any 2 studies, you can expect there to be a high level of heterogeneity between these, caused by different trait measurements, for instance. And in these situations, you have to use meta-analysis in which you combine the two results of the individual studies to not let the sources of heterogeneity confound your results. And the same is true for UGLI and CytoSNP. So the GSA genotypes about 750,000 variants, whereas CytoSNP genotypes only 1,000 SNPs. And that illustrates that you really have to use a meta-analysis for that. And it's a meta-analysis that finds the homogeneous effects across cohorts and sheds light on the heterogeneity and lets you find the results that are the real effects instead of those only influenced by the setup in an individual cohort. METAL it can be used to combine GWAS data, GWAS summary statistics from individual cohorts, and that implements a weighted set score approach. Other methods are also available. Another thing that you want to consider is that 937 samples are genotyped on both platforms, and you ideally exclude these samples in one of the cohorts. We would recommend to do this in the CytoSNP data, and by looking into the sample IDs files of both cohorts, you can find these samples and exclude these from the CytoSNP GWAS. Now I'd like to go into the quality control and downstream analysis after having done any GWAS for any given cohort, or after you've perhaps downloaded summary statistics from the internet. So generally the output of the GWAS is represented as a large table wherein every row represents a variant and the columns indicate to which variant the row pertains, and if that variant is associated to any degree with your outcome measurement. So looking at this summary statistic snippet in more detail, we can see the chromosome position, the variant ID, to alleles, allele frequency, minus allele count, the number of samples included, and beta, the standard error of the beta, and a p-value. And what you generally do when performing follow-up on summary statistics is that you combine your summary statistics to additional data sets, and you combine these with additional data sets based on the RS ID or the combination of genomic location and the reference and alternative allele. You have to keep in mind, however, that summary statistics are not often, or the RS ID in summary statistics are not always included. So in that case, you want to annotate your summary statistics using a database file, for example, from DBSNP. And if you format your summary statistics in VCF format, you can, for instance, use BCF tools, annotate for this. Also keep in mind that your reference and alternative allele can be swapped between the database file that you first ideally have to want to fix using something like fixref. So variance can be in linkage disequilibrium. Meaning that these variants are often inherited together to offspring. So here we have a figure wherein the genomic position is on the x-axis and the log transform p-value is on the y-axis. And this is more or less the common way to visualize genomic associations. And herein we can also see this linkage disequilibrium. So we see the lead SNP and a couple of other variants that are also to a lesser degree associated to the same outcome, because they have a more or less the same p-value. And this is something that and this is an important concept to consider when processing summary statistics, since variants might be tagging the same signal. So for quality control of your summary statistics, You want to exclude variants with a too low minor allele frequency, with a too low minor allele count, and with an information score that is too low. So we want to exclude variants of low allele frequency, since studies are generally underpowered to detect associations for variants with a too low allele frequency, while they do cause performance. And they also make your analysis more prone to genotyping errors. You can exclude variants with a minor allele frequency ideally before performing your GWAS, but could also be done afterwards. An information score that represents how well the variants are imputed, and that's also important to filter on, since a low info score indicates a high probability of imputation errors. Genomic control is a widely used tool to assess if there is inflation of signal across the entire genome. So that's ascertaining whether or not the bulk of the test statistics or p-values are more significant than you would expect by chance. And the concept behind this method is that apart from a small number of SNPs that show a true association with a trait or disease, the test statistics for other SNPs should show no association. Artificial differences in allele frequency due to population stratification, cryptic relatedness, and genotyping errors will affect all SNPs, and this would cause the test statistics to be inflated across the entire genome. So there are a number of ways in which we account for genomic inflation. So, for instance, a quantile plot in which we compare the medians of the observed and expected p-values, so the quantile plot, in which that's a visual inspection, a genomic inflation factor in which we compare the median of the observed and expected p-values, and an LD score regression, in which we take advantage of the assumed difference in correlation of causal variance to LD and the correlation of inflation from cryptic relatedness, etc., to LD. Going into the quantile plot and the genomic inflation factor a bit further. So in this figure we see the an unadjusted model, so a model that's not properly adjusted for cryptic relatedness, etc., and a model for which that is the case. And in the unadjusted model, we see a reasonable inflation of the signal, and this is not the case for the adjusted model. And this is also reflected by the lambda value, which is a 3.2 for the unadjusted model, while it is 1.2 for the adjusted model. And this genomic inflation factor should ideally be between 1 and 1.5, wherein the 1.5 really indicates a high degree of polygenicity, and the lambda value above 1.5 would indicate a highly inflated signal. And this can be easily calculated in R, for instance. Should you expect a high degree of polygenicity, I would highly recommend you to look into the LD score regression method instead. So going into the visualization a bit further of summary statistics, this is an example on the right with the genomic location on the x-axis and the log transform p-value on the y-axis. And every dot here represents a single variant. The red line indicates the significance threshold of 1 times 10 to the power of -8. And we see a number of significant associations. And again, we see the LD peaks as well. So the index variant at the top and a couple of linked variants that also are associated to the same outcome. You can also assess a Manhattan plot based on the what you see. So you should ideally see clear LD peaks, like here, and scattered and isolated points that could represent a rare variance. And in this case, more quality control may be required. You could make Manhattan plots via custom code, for instance, but other tools are available as well, like LocusZoom. LocusZoom is a web application in which you can upload your summary statistics, and then you get a Manhattan plot is generated for you, as is a QQ plot with a lambda as well. What it additionally allows you to do is make genomic regional association plots. So here we see the same again. So the genomic location along the x-axis and the log-to-sort p-value along the y-axis, with an index variance and associated variance around that. And here are a couple of a couple of genes that are located close to the variants of interest. So downstream analysis can entail a number of different things. It all comes down to the integration of additional data. So for instance, one might use the GWAS catalog to see if results are according to earlier findings. And one might also be interested to integrate EQTLs to see if the variants you have found influence expression of particular genes. So in order to report results, you can also apply a filtering step known as clumping or p-value informed LD pruning to find the independent signals. This doesn't identify causal variants per se. In order to do that, you want to look into fine mapping, for instance. So thank you for your attention and for your time. I think we have gone over time by quite some margin. And if there are any questions, now is the time to ask them.
Okay, thank you, Robert, and also thank you, Esteban. So take the mic or put your question in the chat if you have any questions to Esteban or Robert.
I don't see any.
Yeah, Vale, go ahead.
Thank you both for your presentation. My question, well, you recommended, of course, to exclude the overlaps between the CytoSNP and the GSA data, but because I already did a GWAS based on these data sets, I also excluded from the CytoSNP those first-degree relatives, so to, avoid unnecessary. Well, to have more independent samples. So, what's your advice? Do you really recommend only to exclude only the exact overlap, or also to consider to remove these first-degree relatives?
So I would say that your approach would actually be an optimal approach, or at least more optimal than not excluding first-degree relatives. What do you say on that, Esteban?
Yeah, so I think that I think I forgot to mention. So of course you should remove relatives, but that would cost you sample size, right? So I would recommend more to include them, but in the framework of a linear mixed model. But I mean, both approaches are accepted. It's just that it will cost you sample size if you remove them.
Yeah, but I think Vale is referring to the relatives between the cytochip and GSA cohort.
Oh, sorry, I was not. Yeah, sorry, I didn't understand that. Yeah. It's correct then. Yeah.
Yeah, because then you want to meta-analyze them. You have to remove all dependent individuals, otherwise your results may be biased.
Yeah.
Thanks.
Yeah, so remove, and then preferably remove them from the cytochip, because that is the worst chip with a lower number of SNPs. Okay, thanks, Vale. Other questions?
Yeah, thank you for your addition.
Then thanks more, Esteban and Robert, for this clear and nice overview of genetic analysis GWAS in Lifelines on the high performance cluster. And then I suggest to have a 10-minute break till 1:45, and then we'll continue with the next lecture.
Thank you very much.
Okay, thank you.
Bye-bye.
Thank you very much.